Hi everyone, welcome to this week's AP Bio Chat. Today we will be discussing two biotechnology techniques, and we'll be discussing them in the context of some free response questions that might resemble something you might see on exam day. For those of you who haven't joined us before, you can look at all of the live programming that the DNA Learning Center is providing for students and for teachers. We know that learning from home can present quite a few challenges, especially as it pertains to DNA science. And so we're hoping that some of these resources might help you. Feel free to check out our schedule of programming. There are things like live labs, museum tours, all happening, so check it out. And to keep in touch with us about what types of programming we're doing, please feel free to follow us on our different social media channels. I also just like to remind students that these are the topics that are going to be on this abbreviated form of the AP Bio exam. This is only for this year. So everything from your biochemistry and organic chemistry up until gene regulation and expression will be covered. That includes the biotechnology things that we're doing today. And then if you haven't seen this already from the AP exam writers, for test security purposes this year only, everyone will be taking the exam at the exact same time globally. This will prevent students from sending each other questions and enhance testing security and make sure that the test is fair for everyone. It's open book and open note. And you have one 25-minute long free response question, which is lab-based, and one 15-minute long question that is concept-based. So this should sort of frame your studying and your strategy for studying from here on out. And then today we're going to be talking about electrophoresis and PCR. Our website has really great animations that review these concepts. I'm going to go in like very bare bone minimalistic style detail. So I'll go through those slides with you in a minute, but if you do want more detail, you can check out our website here. It's dnalc.csh.edu/resources/animations. And there are animations for all of the biotechnology experiments that you might want to study before your exam. All right, so we're gonna start with a sample free response question. I think this is reminiscent of maybe what you'll see for the second free response, where it's not lab or data-based. Instead, it's just concept-based, right? And the concepts that are being covered here are, I'd say multifold. So it says, describe how polymerase chain reaction can be used to make copies of a gene of interest, explain the significance of using various temperatures during PCR, compare the laboratory technique of PCR to in vivo DNA replication. For those of you who haven't seen this word before, it is an expression that might crop up on the AP exam. So in vivo just means in a living thing. So when we're talking about experiments in vivo, we're talking about experiments that are being done inside cells, inside organisms, right? The sort of opposite of in vivo would be in vitro, which I think we'll see a little bit later in the slides. Okay, so you should know the difference between in vivo and in vitro. And I say that this question is multifaceted because PCR in and of itself is multifaceted. So let's talk a little bit about that and then we'll revisit this question. All right, so what is PCR? It's polymerase chain reaction. It's used for in vitro DNA replication, meaning we're replicating DNA in a test tube. PCR is a specific process. This is important. So unlike in vivo replication that happens in your cells, in vitro PCR is specific in that we are only replicating a gene or genes of interest. So we can actually design these things called primers that act as little search tools and just zoom in on the gene of interest. So that way you're not making replicates of your entire genome or your entire DNA sample, but instead just making copies of a single gene or a single piece of DNA. Similarly to in vivo replication, the replication process results in exponential growth, meaning Every DNA strand is going to, when it's replicated via PCR, it's going to result in double the amount of DNA. So you start with one copy of your gene. After one cycle of PCR, you end up with two copies. After your second cycle, those two double and become four. After your third cycle, those four double and become eight, and so on and so forth. How do we model this exponential growth? This is something that might show up, but just a quick review here. The number of copies of DNA that you would end up with is equal to 2 to the n power, where n is the number of cycles that you run. So this is something that you can control as the experiment designer. Usually PCR reactions, you see them running around 30 cycles. And so you end up with 2 to the 30th power copies of DNA, which is on the order of billions of copies of DNA. The tool used to complete PCR is called a thermocycler. This sounds fancy, but thermo, we're just talking about heat, temperatures, and cycle means that you're repeating things, right? And so we're going to talk about exactly what kind of alternating of temperature is going to happen, but you're basically switching between heating up your samples and then cooling them down. And we'll talk about why that is. This is kind of important. So it utilizes similar reagents as in vivo replication. So we talked about in vivo replication in one of our earlier AP Bio Chats. But remember that replication requires enzymes, and so does in vitro replication. So in vitro replication, we still use a DNA polymerase. Remember, DNA polymerase is the enzyme that we use to catalyze the addition of nucleotides to a forming DNA strand. But because we are heating up our samples, we need an enzyme that is thermostable. If you guys remember from your unit on enzymes, enzymes are actually just proteins, and proteins are folded together, and their folds are mediated by hydrogen bonds. Hydrogen bonds are pretty weak, and so you can change the temperature or heat up proteins and cause them to unfold or denature. So we need an enzyme that is able to resist those types of heat changes because we are going to be heating up our samples. And so we use an enzyme called Taq polymerase. Taq polymerase is actually an enzyme that is derived from a thermophilic bacteria. And so thermophilic bacteria is a type of bacteria that thrives in heat. And so we use a Taq polymerase instead of a human polymerase. But the other sort of ingredients or reagents are pretty familiar to us, right? We've got some primers. Primers are going to be used to sort of initiate synthesis of genes. And the primers are designed ahead of your experiment. So you can design a primer that is specific to a certain gene based on studying the sequence ahead of time. Then you need nucleotides. Remember, these are the building blocks of DNA. And then finally, you need buffer. And in any experiment, you probably will be using a buffer. Buffer is just a fancy science word for a solution that helps keep conditions stable. And in this particular case, the buffer is also supplying ions that the enzyme needs in order to work, or what we would call cofactors. All right, any questions so far? That was a lot. So what PCR is doing is we're making copies of DNA. It's doing it in an exponential fashion. It takes advantage of changing between hot and cool temperatures. And it uses reagents that are pretty similar to in vivo replication, with the exception of this Taq polymerase being able to withstand heat. All right. So I'm going to go through the different temperature cycles that you would typically see in a PCR reaction. So the first step is to do the same thing as you would in vivo. We need to separate our DNA molecules. In vivo, we separate DNA molecules using enzymes. In PCR, we separate DNA molecules by denaturing them. In other words, we break them apart by using heat. So the first step is heating up your reaction. You'll usually see the first step of a PCR reaction being run at around 100 degrees Celsius. Remember that the bonds that are holding together our strands of DNA are weak. They're hydrogen bonds, right? And so when we apply this heat, the double-stranded DNA molecules are going to break apart, shown here in this cartoon. All right, so we started with our double-stranded DNA, we apply heat, and our molecule splits apart. The next step is adding primers. And this is really similar to what's happening inside our cells too, right? We remember that RNA polymerase lays down some primers and allows synthesis to start, right? The same sort of thing happens here, but in order for the primers to stick, we need to cool the temperature down, right? Because just from a thermodynamic standpoint, if things are really hot, molecules move really fast, and we're not likely to have those hydrogen bonds being formed. So we need to cool down the reaction so these primers can stick to our template strands. And so we usually cool the reaction down to about 50 to 60 degrees Celsius. Um, and I've indicated the little primers here with these green lines. So the primers are going to stick to both the template strands. And this is going to indicate to the polymerase where to add nucleotides. So that way we're only making copies of our gene of interest. And then similar to what's going on in your cells, our last step is extension. Extension is exactly what it sounds like. It is the extending or elongating of the gene, right? And so we're using in this step DNA polymerase. In this particular case, we're using Taq polymerase to add to our template strands and complete gene replication. To do this step, we usually heat up the reaction slightly. So it's usually going to take place at around 70 degrees Celsius. The reason we warm up the reaction a little bit is just to increase the speed of addition of nucleotides. However, you'll notice that it's not as hot as the first step. All right, so going back to our free response question, remember the strategy that I gave you guys a few sessions ago is to break down the question and make sure you're really addressing each one. So I see a couple of logical breaks here, and you don't have to overthink this. The logical breaks for me are just where the periods are, right? So the first task I need to do is describe PCR and how we're using it to make a gene of interest. The next break for me is explaining the different temperatures and why they're significant. And then the last break for me, when I'm sort of organizing my answer, is to compare PCR to in vivo replication. So there are three tasks really that I need to focus on for my answer, right? And if you're tuning in live, we're going to go through the answers right now. But if you're streaming this at a later time, now is a great time to pause the video, set a timer for yourself for 15 minutes, and try and answer this question given the review that we just did together. All right, so I'm going to give everyone just a second here to think. All right, and then this is what I would do. So I would start again with these first two chunks. So the first chunk just says, describe how polymerase chain reaction can be used to make copies of a gene. So all I'm going to do here is talk about what reagents we're using and what methods we're using, right? So very simple. I just said PCR replicates a gene of interest using special primers, nucleotides, DNA polymerase, and alternating heating and cooling cycles, right? That's really all you need here. So you don't really have to do more than what the question is asking you. You'll notice, as I indicated before, it's really useful to demonstrate that you have a solid command of the vocabulary. So notice I'm using words like gene, primers, nucleotides, polymerase. These are all going to be really important for you to get as many points as possible. All right, so for part two of this question, you're going to have to go into more detail. So you effectively are going to have to explain what is happening at each of those three steps that I just described. So you're going to need to describe the denaturing step, the annealing step, and the extension step. And you're going to need to explain what is happening to the molecules. And I would try and use the vocabulary here. So try and know the words denature, annealing, and extension. I'm not going to read my answer to you because I know that you guys can all read. So if you are watching this live, you can feel free to pause the video. And if you're streaming it at a later time, do the same and give yourself a little score for how you would have performed on this part so far. For the last part, it says to compare the laboratory technique of PCR to in vivo DNA replication. So the main differences, right, are that PCR is temperature controlled, whereas DNA in vivo replication is more enzyme mediated. They're both going to also be using an enzyme called polymerase, um, but the polymerase that we're using in PCR is a type of polymerase that is heat resistant. So those are going to be the main differences. Um, and so you would just focus on those. And again, I'm not going to read the answer to you. Feel free to read it on your own and reflect on how you would have answered this question. All right, and then for our wrap up of today's session, I wanted to give you guys a sample of what an electrophoresis free response question might have looked like. So this would be an example of a long question. So the 25-minute one that is based on an experiment, right? And I did kind of abbreviate the text here just because there were a lot of components to this particular question. So what I'm going to do is I'm actually not going to go through the answers to this one today. We'll go through the answers for this one on Thursday. This will give you an opportunity to try this one out for yourself and then tune in with us next week so that we can go through the answers together. But I am going to go through very briefly what electrophoresis is and sort of how to start thinking about this particular question. So there are a lot of concepts that are biotechnology based that are kind of built into this. So the first one, whenever you see like a map of DNA like this, and this one happens to be a linear map, but you might also on the AP see a circular map because remember, some organisms store their DNA in a circular loop, such as bacteria, viruses, right? This is a linear strand of DNA, so I think it's a little bit easier to sort of look at. The only reason I didn't do a circular piece is because I didn't have a ton of space on this slide. So just be aware that you might see a circular map or like a plasmid map. In any case, the concepts are the same, right? So they're telling us this restriction map shows the restriction sites for two enzymes, enzyme A and enzyme B, on a DNA fragment with a total length of 5,000 base pairs. All right, looking at this map, are we looking at the entire segment of DNA on this map? Like probably not, right? Because this number here is 1,600 and it says it's 5,000 base pairs long. So we're probably only getting a partial map. So that's first thing to recognize. The second sort of concept that you guys should know here are restriction enzymes. For those of you who need a refresher on what a restriction enzyme is, a restriction enzyme is just an enzyme that cuts DNA at specific restriction, what we call restriction sites. Okay. And each restriction enzyme is going to have a specific restriction site. So those are the concepts you need to know about restriction enzymes. And so this first enzyme is cutting the DNA here. It recognizes some sort of sequence here. Maybe it's ATGC, I don't know. And whenever it sees ATGC, it cuts, right? The other thing you need to know about restriction enzymes is that they produce these things called sticky ends. So remember, your teacher might have drawn you a diagram that looks kind of like this, where you have a piece of DNA that looks like this. The restriction enzyme will cut like here, and therefore produce these like hanging over pieces that can stick together, right? I like to think of it in terms of Legos. So if you had two Legos that were just rectangles like this, you couldn't get those Legos to stick together. But if you built two Lego pieces that looked like this, and then like this, you could get these pieces of Legos to connect, right? So that's what sticky ends are. They're basically these jagged ends that allow you to glue pieces of DNA together. In any case, that is what you need to know about restriction enzymes. I'm gonna clear these drawings and get back to the question. So these two enzymes are cutting the DNA at these different sites. The first question chunk tells us to explain how gel electrophoresis separates DNA fragments. The second asks us to describe the results and tells us that we can draw a diagram. So I think that if you draw a diagram, I would encourage it because I feel like it helps you understand the problem better and keeps you organized. However, grader wise, if you are doing a diagram, you need at least a sentence to explain what each diagram is showing. So it's not a diagram or like a diagram is not a way to get out of writing things. So for those of you who are afraid of writing, diagram is not your free pass. You still have to explain what your diagram is showing. So they show us 4 scenarios. So they want us to explain the results you'd expect if you digest with only enzyme A, digest with only enzyme B, digest with both enzymes, and then don't digest. Digest just means use restriction enzymes to cut DNA. In biology, anytime you see the word digest, it just means break apart. Predict, and then the last part says predict how the results would be different if a mutation occurred at the restriction site for enzyme B. So if there were a mutation in the DNA here, how would the results be different? Okay, it's a lot. And this is why this is a good example of the 25-minute question. In any case, let's do a quick review of gel electrophoresis. Electrophoresis is basically a tool that allows us to visualize a sample of DNA that we have. It separates DNA based on size and charge, and it utilizes a gel matrix, which is just a fancy sort of, I think of it as the kitchen tool, you know, like a sifter or a sieve that we use to separate big chunks of flour from finer particles of flour that we want to use in our baked goods. Or that we would use to separate out noodles from a pot of boiling water. The gel matrix is the same thing. It's a sifter. The only difference is that we can't see the little mesh holes with our eyes. And so this particular sifter is good for separating really tiny molecules like DNA. All right, so this is a basic schematic. You should not be afraid of an overview or an overhead bird's eye view of a gel. This is pretty typical. You might see this on the AP. They might have you interpret a gel. In this particular case, they're asking you to sort of interpret results without seeing the gel. In any case, these little rectangles at the top are where you would put your DNA sample. They will not draw, at least I don't think they will. They probably won't draw the negative and positive ends. But when you run electrophoresis, electro means, you know, electricity, we apply an electric current to the side the DNA is on. Electricity is just a flow of electrons. And as you probably learned in another science class, electrons are negatively charged. What else is negatively charged? DNA. And so when we apply a negative charge to the side that we are loading our DNA on, the DNA is going to be repelled by the electric charge. Another way of thinking of this is that applying a negative charge to this side creates an electric gradient, right? You should remember the word gradient from earlier units. Gradient just means a difference, right? So when you apply a negative charge here, the charge overall becomes very negative on this side. These are all my negative signs. And so the gradient that results means that by contrast, this side is going to be relatively positive, right? And so another way of thinking about this is that the DNA is going to be attracted to the positive side of the gel, right? And so which direction will the DNA move? The DNA will move towards the positive end, right? That makes sense. So DNA again is negatively charged. If you don't remember why it's negatively charged, it's because of the phosphate groups. So they're going to move down the gel towards the positively charged side because opposite charges attract. And then the sort of last concept you need to remember for electrophoresis is that smaller molecules are going to move further in the gel. Larger molecules are going to get stuck back in the gel. So if you see a molecule that is closer to where you originally put it, it's larger. If you see that the molecules have moved further in the gel, they are smaller pieces of DNA. And we can use a tool called a ladder, which is basically like pre-digested DNA. It's your positive control with known fragment sizes to determine how big in base pairs your molecules are. Okay, so this is just a general schematic. If you have trouble remembering that larger fragments get stuck back and smaller fragments move further, a nice helpful analogy is thinking of a traffic jam. If you are stuck in traffic and you are on your school bus, right? Um, I'm sure all of us are kind of longing for the next time that we can go on a bus. But in any case, if you're stuck in traffic on a bus, you probably aren't going to have a lot of luck getting out of traffic anytime soon. Why? Because a larger vehicle is really difficult to maneuver through tightly packed traffic. Um, if you, however, are riding your motorcycle, right, you are able to weave in and out of the crowd because you're smaller. You can get through those tightly packed crevices of cars, right? The same sort of idea applies here, except instead of a traffic jam, you've got this like gel matrix, right? Okay. And so I'm going to leave you. Oh, let's clear these drawings. I'm going to leave you with this question. So what we'll do is next time we will start the session by going over this free response question. So what I would do is if I were watching this, I would just pause the video or screenshot this problem, try solving it, and give yourself 25 minutes to mimic test day conditions. Um, Whenever possible, you kind of want to make sure you're preparing for a test as close to test conditions as possible. So give yourself 25 minutes to do this, and we will go over this next time. Just for your information, next week we will only be doing one of these AP Bio chats. So we're cutting the Tuesday afternoon session. We're going to do a Thursday at 2 p.m. So I'll meet you here in a week. So we'll go over the electrophoresis free response question. I also wanted to talk a little bit about gene regulation. So I know that that's a really tough concept for students. We've already talked about transcription and translation. So next week, what we'll do is we'll talk about things like promoters and operons and how those work to sort of turn on and off genes and why we need those types of systems in our cells. In the meantime, if you have any questions, feel free to send them to us on Instagram. Our handle is DNA_learning_center. I hope this was helpful. I hope you are all healthy and well, and I hope that your AP preparations are going well. Thanks for joining us today. Bye.